much. So, good morning, everyone, and thank you very much for joining with us today. Today, we have a talk regarding the algae, and then let me take a minute to give a brief introduction to our guest speaker today. She is Dr. Renu Geetha, and she did her PhD in Chemical and Environmental Engineering from the University of Nottingham. Currently, she works postdoctoral researcher at the Estonian University of Life Sciences and also teaches at Tallinn University, Tallinn, Estonia. Her current research focuses on the development of high-value biomaterials from algae resources. So Dr. Geetha's research interests are biotechnology, nanotechnology, marine algae biomolecules, nanomedicine, anti-cancer drug discovery, drug delivery, nanotheranostics, carbon nanomaterials, liposomes, microbiology, molecular biology, and cell culture techniques. For today's talk, she's going to talk about algae, its potential in food, agriculture, and biomedical applications. Dr. Geetha, please go ahead.
Hi. Hello, everyone. I'm Renu Geetha. I'm a senior researcher currently working in Estonian University of Life Sciences. I started my algal research back in 2008, where I did a bachelor's project. It was focusing on the production of biofuel from microalgae. I used a strain called nanofluorosis, and we were trying to extract biofuel out of nanofluorosis. Then later on, I moved to nanotechnology. Then the research focus was changed, and later in 2019, I came back to algal field again with Dr. Renu team in Tallinn University, where we were focusing on macroalgae, various macroalgae and their bioactive compounds and their bioactive properties. And currently, I'm working on microalgal field, and it's an interesting, very exciting journey. And I would like to share some of the our research experiences and some general topics about algae. What are the future potential and the current potential of algae in food, agriculture, and biomedical applications. This is kind of a very basic lecture only, not very focused on. So I hope most of the listeners will be quite new to the algae field. So I made it like simple for you to understand it quickly.
Sorry, Geetha. Sorry to disturb you. Can you make it presenter mode? We still can see your normal thing.
Is it better now?
Oh yeah, that's fine. That's fine now. Okay. Thank you.
So before moving into the algae field, I would like to talk a little bit about blue biotechnology, which is a part of blue bioeconomy by Europe's one of the major strategies to offer solutions to produce materials, enzymes, food supplements, and pharmaceuticals. So basically, this field of science is using renewable aquatic biological resources such as macro and microalgae, bacteria, fungi, and invertebrates. So basically, they were used for food additives, animal feed stuff, pharmaceuticals, cosmetics, and energy applications. So according to the major biomass resources in this blue biotechnology comes with micro and macro algae. So basically, algae is classified as two different based on the size as microalgae and macroalgae. Most of the microalgae are mostly unicellular in nature, and they are microscopic. And we still have like some macroscopic microalgae as well. Then it comes with the dinoflagellates, diatoms, blue-green algae, golden brown algae, this type of different varieties as well. And they are also referred, also the phytoplanktons comes under the microalgae category, and they are larger in population, and they are considered as a superior food source currently. And it is a food source for soil microbiome to improve the soil quality. Where microalgae are multicellular, they could be few meters long, as you can see it in the picture. The brown algae kelp, they could be a few meters long, and this will be some of the pictures you might have seen in TVs or like in some other media. Then macroalgae are mainly differentiated based on their pigment as red, green, and brown algae, and they are referred to as seaweed. So here, this is one of the brown algae. You can see this is like probably few meters long from the seabed, and they are like good carbon fixation agents. They help to maintain the carbon footprint to a better level at the moment. So this is a large picture of this same sargassum macroalgae species. And this is how the aerial view of an algae populated water stream will look like. And for someone who doesn't know algae at all, this is the green slimy thing which we see in any water bodies. So as you can see in the rightmost picture. So basically microalgae will look like the leftmost picture with a single cell. Sometimes they will be joined together with a cell wall, agglomerated together with cell wall because of the presence of polysaccharides. And this is how it looks like. So algae types and applications. So as I mentioned, based on the pigment, algae are differentiated as red algae, brown algae, and green algae. Algae actually have like very variety kind of classifications, and it is still updating. So according to one of the latest article, it has like 11 different classification, and more and more new algae are like identifying. So this classification is very general based on the pigments of it. So if you search for the algal classification types, you can find like hundreds of different types of classifications. Among that, it will be differentiated based on which kingdom it belongs to, based on which pigments it belongs to, based on the size, based on many other properties as well. So the major applications in algae are in food, agriculture, environmental applications, biomedical applications, energy applications. All of these applications are interconnected as well, most of them. So once you think about the environmental application, this is the major point where it has a good role in carbon dioxide sequestration. So Europe's 2035 strategy is to make European nation a climate neutral society. So algae as a renewable source has a good part in this carbon sequestration, and as a renewable source, it promotes the sequestration by carbon absorption and carbon fixation. So this has a major role, and they are a major part in wastewater treatment in the current scenario because of the absorption capacity of heavy metals and various other synthetic industrial byproducts. When it comes to the food applications, these are used in animal feeds, then food colorants, because most of the colors used in food nowadays are synthetic colors. So these could substitute the food colorants. Then they could improve the quality, corrosion remover, flavor supplement. These are the food applications, and biomedical applications comes under the same category like pigments, polyphenols. Then they are providers of DHA and EPA. Their antioxidant potential is really high. Then polyphenol, then biostimulant and nutraceutical properties. All of this will be explained again later in detail. So when we are talking about microalgae, they're photosynthetic unicellular organism mostly, and they grow rapidly, and they're a good source of protein, lipid, carbohydrates, and pigments. And the good thing is they are easy to cultivate. Still, it depends on the environmental conditions and the required media and other conditions, and they are a renewable resource. That's the best part. So if you provide the best conditions, they are renewable. So that's the beauty of this algae field. So as you can see, this will be a basic lab setup where we will be able to cultivate microalgae from a single cell. So these are different microalgal strains with a different morphology. They could be single cell, filamentous, needle shape, spherical, and these are like single cells attached together. So they occur in different sizes, different shapes. So why algae is more popular these days? Algae is a very sustainable source for different products. So they are used in food supplements, cosmetics, biofuels for energy purposes, animal feed, plastics, textiles, fertilizers, and medical applications. So if you think like you have never used the algal products, probably there could be algal products in your paste, or maybe in your chocolate, or maybe in your detergent also. So it's that common in these days. So most of the food products these days contains some of the stabilizers, which could be agar or carrageenan, which are algal polysaccharides. So it's very common to use algal products in food applications these days. How do we cultivate algae? As I mentioned before, this is mostly explained about microalgal cultivation. So in the case of microalgae, we can actually start the culture from even a single cell. So basically, same as microbial culture, we will start from the seed culture, and we can we can streak it into a agar plate, and from that we can transfer them to small conical flasks, like 25 ml or something, with the proper media. Each algae has its corresponding media, which will provide the proper nutrients and proper growing conditions for them. So from the agar plates, it could be transferred to bigger flasks. Then it could be transferred to photobioreactors. Photobioreactors are where all the conditions are maintained: the pH, salinity, gas intake. All these conditions will be maintained to get the suitable products out of it. So based on what are the end products we require, we can engineer the growth and the production conditions in a photobioreactor. Also, since they are photosynthetic organisms, they need light supply. So basically, we can provide the specific light sources based on the requirement as well. Light source and light intensity, then light period, how long it needs light. Some of the algae needs stress condition. When the light is not there, they will be stressed and they could change color. For example, one species called Algae, they normally grow in green color. Based on the nutrient condition, based on the stress environment, it could change color and the pigment will be producing yellow color. Similar to that, another strain called Haematococcus. Haematococcus, one of the figures here, you can see like a purple color. So normally, Hematococcus in its vegetative condition will be green in color, but when it is under stress, it will create a red pigment called astaxanthin, and that is red in color, and the entire culture will turn into red color. So based on the conditions, growth conditions and situations, the pigment production, the protein production, fatty acid production, all these scenarios will change. So in a lab condition, we could grow it in photobioreactors with extremely strict controlled conditions. And if it is grown in a large population way, it will be cultivated as open ponds, as you see in the figure, open pond systems, or like in closed system itself, but in like tubular systems. So these are the different cultivation systems where algae could be, microalgae could be grown. So food applications. As I explained before, algae are rich in proteins, amino acids, fatty acids, carbohydrates, vitamins, and minerals. So they are a good source of all these different essential products. And they are source of various other bioactive compounds as well. Then they are rich in polyphenols. Polyphenols are good antioxidant compounds, so they provide excellent nutraceutical application. Then, as we mentioned before, pigments. Most of the microalgae are green in color and they are rich in chlorophyll content. Some of them have like other colors. They include keratin, astaxanthin, erythrophytin, different pigments will be there. So these pigments could be used as colorants, safe, natural colorants for different food applications. Then the uses in food industry. First one, as we mentioned now, like green safe pigments as colorants. So basically, chlorella itself is produced in yellow color, white color, and green color pigments with the symbol mutations. So these colors are safer than the synthetic colors, so it could be used in various food products without much health concerns. Then they are used in meat as emulsifier, meat, dairy, bakery, beverages, all these different products use microalgae, different contents of microalgae and macroalgae. As dietary fiber and a mineral source, they are used. They are used as emulsifier, gelling agent. They are also like a good lipid oxidation preventing agents. Then, since they are rich in proteins, vegan preferred alternative food for proteins could be used this for food. And nutraceutical products and feed preparation. Another important aspect is feed preparation for fish feed, poultry applications and all. You could use this. Next application is in agriculture. In agriculture, it is used as a biopesticide. So for weed management and for different resisting different crop pathogens because of its antimicrobial, nematocidal, herbicidal, and insecticidal properties. It is used as a biopesticide, and it is also used as a biostimulant where it could improve the plant metabolism and enhance the growth, where it could uptake nutrients and it could resist the environmental stress to an extent. Also, it is a very good bioremediation tool where it can absorb heavy metals, synthetic dyes, pharmaceutical and personal care product byproducts. All these different industrial anthropogenic byproducts could be absorbed by microalgae, where the water could be used as a water, or the soil could be used as a safe environment. The major role is natural soil enhancer, where from ages we are using micro or macro algae as a green sustainable fertilizer. It is a solution to human health, where so many concerns are raised from chemical fertilizers like cancer and hypoxia conditions. And it is a solution to environmental problems like eutrophication and erosion that's also caused by chemical fertilizers. So in agriculture, it is used for ages, actually, the green sustainable fertilizers. But at the moment, the uh, biostimulant properties are really utilizing, where we will be focusing on various growth promoters, various enzymes from microalgae, which could promote the growth of various plants. Biomedical potential of algae. So in biomedical potential, the first point is fluorescent tagging. Here also the same scenario of pigments comes, where phytoerythrin, for example, it is a very fluorescent purple color pigment. This could be utilized in fluorescent tagging, where the drug carriers or the carrier systems could be like easily located in different infectious areas. So this could be used in fluorescent tagging in different medical applications, and it could be used in tissue engineering. The various agar or carrageenan-like substances are used in the production of hydrogels, which could be used as a scaffold material for tissue engineering purpose. So basically, instead of the normal cytosine scaffold, which are used in tissue engineering, we could be incorporating algal-based polysaccharides or algal-based biomass as a tissue engineering scaffold material. So it could replace the existing biomaterials default demerits. Actually. So, for example, like if all right, let's go to the next point. Actually, so antioxidant potential. So antioxidant potential, as I mentioned, polyphenols, polyphenolic content of micro or macroalgae corresponds to the antioxidant potential of the microalgae, and they are used in various safe cosmetic applications, cosmetics, and also dermal applications of skin therapeutic applications as well. So from the various bioactive molecules which are providing proper antioxidant potential, this could be used in skin cancer. This could be used in various skin conditions where skin tissue engineering is also benefited. Then another new very important topic is like the drug delivery carriers, where this algae could be used as because of the microalgae condition, single cell condition, they could be used as a proper carrier for the delivery of various drugs or other therapeutic materials. For example, extracellular vesicles are some of the products which are like excreted from the algal cells. They contain nuclear material which could be utilized for carrying various therapeutic materials or various drugs for different properties. So there are reports where they are used in antimicrobial properties. So these are an excellent, safe, and sustainable drug carrier system as well. There are multiple reports on the antiviral, anti-cancer, and anti-inflammatory properties of microalgae and macroalgae, where it also showed different immunomodulation properties, where immune cells are supported by the presence of these natural products. The figure below is the picture of Haematococcus, which I explained before. The first figure is Haematococcus under stress condition where it turns to purple with the pigment as a sample. The other two pictures are the vegetative stages where they are still in green color with the chlorophyll. So these are the biomedical potential of micro or macroalgae. So algal bioactive compounds. This is one example of how algal-derived pharmaceuticals are acting as an anti-cancer agent. So as you can see here, various algal components such as blue-green algae, green algae, brown algae, red algae, golden brown algae, all of them are having like different bioactive compounds. So based on the bioactive compound from each algae, they could be used for targeted cancer therapies. So as you can see here, for example, from brown algae, we can extract ficoidam, ficosanthine, pheophytin, ascophyllin. All these are different polysaccharides extracted from brown algae, and this could be utilized for targeted cancer therapy for bronchopulmonary cancer, prostate cancer, lung cancer, breast cancer. Similarly, from each of the other algal strains, there are different bioactive compounds which could be used for various targeted cancer therapy. And another another field of algal application is in the field of bioenergy, where algae is a very good source of biodiesel, biobutanol, bioethanol, methanol, biogas, biochar, bio-oil, and biohydrogen. Biohydrogen is very important these days because of the clean energy concept of those. And they also contribute to microbial fuel cell based bioenergy as well. So all this has the advantage of being clean energy producers and a renewable energy source. So energy sustainability goals will be could be addressed in near future using more of the algal resources. And how Estonia is doing with the algal resources. I am explaining only about like some of the macroalgal resources because microalgal resources we are still like identifying most of the species and strains. So these are some of the macroalgal resources we have in Estonia. And I'll be talking about one strain called Fusariella lumbricalis. And this is found in Kasari Bay, which is a small location near to Saaremaa in Estonia and within Estonia and Finland. And these are the pictures of this various macroalgal resources found in Estonia. This pointed location is Kasari Bay, from where this Fusariella lumbricalis is extracted. So Fusariella lumbricalis produce an anionic partly sulfated polysaccharide called fulsaran, and this is classified with the carrageenan, and it complies with the EU directive and Food Chemical Codex and GCFA standards, which corresponding to the food quality of products in Europe. So it's a safe food product. It is classified as a safe food product. And Estonian is the company which was the only company producing Fursuran before. I don't know like whether there is any other company now. They are located in Estonia, and they produce Fursuran from 1966. And this is the hydrocolloid, and it has about 75% with weight. And this is the process where they are acquiring this Pulsalaria algae, macroalgae from Kasari Bay area. This is the red color algae, and the pigment is phycoerythrin, and it's a very fluorescent pigment. This is of very high importance and very high value based on the purity, and it is used in fluorescent tagging, and it's also used as colorant in cosmetic applications. So these are some of the uses of Fursuran, where for food application, it's used in dairy products. Basically, the polysaccharide will be extracted from the Fursuran by either ethanol extraction or hot water extraction process, and it is used in food, non-food, and medical application. In food application, it could be used in dairy products, beverage, clarifying agent, nutritional supplement, edible coating, and bioplastic. And in non-food application, it is used in personal care products, coatings, and makeup, cosmetic applications. Then in medical scenario, it is used as fluorescent tagging agent in biochemistry and microbiology, and it's also used as a cell immobilization agent, and for drug delivery application as a safe drug carrier, and a tissue engineering application as a scaffold, and molecular biology as a carrier system and a gel system. So now I'll be discussing a little bit about our research in Estonian University of Life Sciences, where we will be dealing with different microalgal strains. And at the moment, I am focusing on one particular strain, Chlorella vulgaris, and it is used. It will be cultivated in presence of flue gas. So the focus of our project is waste or byproduct utilization for microalgal growth by promoting zero waste philosophy of this circular economy. So to reduce the carbon footprint, this greenhouse gases has like a good role in increasing the carbon footprint. So in our research, we are focusing on utilizing flue gas as a carbon source for the growth of microalgae. We are using Chlorella vulgaris here, and introducing flue gas as a carbon resource. So we have like a small furnace facility in EMU where from the chimney the flue gas will be collected, and this will be utilized as the carbon source in our cultivation system. So in the photobioreactor, instead of carbon dioxide, we will be producing the flue gas, where the carbon tolerance of each strains are tested, the growth conditions are tested, and based on that, the tolerant strains will be cultivated with the specified optimized conditions. So the flue gas treated chlorella will be cultivated initially in smaller volumes, and then it will be upgraded to the large volume photobioreactors. We have like a 100-liter, 300-liter facility photobioreactor here. So after the mass cultivation, chlorella will be harvested. In chlorella or any microalgae will be harvested, and then the product will be subjected to different biomolecular analysis, and the dried powder will be used for the production of fish feed, aqua feed, for the salmon or trout rainbow trout fish. So after the growth condition, protein, carbohydrate, lipid, pigment analysis will be done with the presence of low gas, and without the presence to analysis the safety and to analyze whether there is any harmful products are accumulating in the microalgae. And after the safety evaluation, it will be used for the fish feed production, and it will be used as a feed for the different fishes. So chlorella, Senedesmus, Haematococcus, all these different strains are used for this fish feed production facility. And currently we are focusing on chlorella. And this is a ongoing project. And the next project in our team is based on utilization of another side stream, where agriculture waste from wood industry is used as a growing material for microalgae. Here, the byproduct, the condensate of torrefaction is a high temperature thermal process, and the byproduct of torrefaction as torrefaction condensate is used for the algal cultivation. And this is used for the production of exopolysaccharides. So basically, we are using exopolysaccharide producing microalgae and growing them in torrefaction condensate present media. So basically, torrefaction condensate is a waste stream which will be discarded after the torrefaction process. So we are utilizing this waste stream as a nutrient source for the production of algae, and the final product will be exopolysaccharide, and this also will be utilized in various biomedical applications and for production of surfactants and for the production of for the production of surfactants and some other economic value products. So we are focusing on the first part of this at the moment, where the torrefaction condensate from different temperature regimes are used for the cultivation of different microalgae, and the exopolysaccharides which are present in the media are extracted and recovered and used for different evaluations, like various biomolecular evaluations. So when we are talking about algae is actually future tool where we could obtain the UNESCO Sustainable Development Goals, most of UNESCO Sustainable Development Goals, where this would contribute to about 13 different goals of sustainability. This, if it is used properly, this could be an alternate source for like addressing many other scenarios as well. So as you can see, this promotes good health and well-being. It supports cleaner water and sanitation. Wastewater treatment is another very important aspect of microalgae and macroalgae, and it creates clean energy, and it creates it helps to reduce the carbon footprint, and it provides industry and innovation and infrastructure. Also, it supports sustainable cities and sustainable communities, and it helps the reuse of various industrial side streams to use as a nutrient source for production of various high value molecules or like low value molecules like fertilizers and it has a major role in climate action, and it improves the life below water and life on air, and it supports the partnerships for the goals as well. So these are the references I used in this presentation. And I would like to acknowledge Professor Timotheus and my colleagues at EMU and Dr. Vikani and the team at TLU and Blue Bio project, Iran Horizon project, which is called Microalgae in IT, and Professor Alireza and team from Pivot team at the EMU. Also, Dr. Dulmini for the invite, and Sebastian for coordinating. Thank you so much.
Thank you very much, Geetha. It's really interesting and detailed talk for every one of us because it's quite new. This algae topic is quite new to most of us, I guess. Thank you very much. So now the floor is open for questions.
Yeah.
Yes, please go ahead. Yes, Gurat, please go ahead.
Oh, sorry. Can you hear me?
Yes, yes.
Yeah, okay. Thank you for a very interesting presentation. I don't know much about the algae field, so I have like a couple of questions here. So you talked about that we can use them as scaffolds in the tissue engineering. So I wanted to know how stable are these? How stable they act as scaffolds? Is there any research ongoing with that about like these scaffolds?
Okay. Yeah, as tissue engineering scaffolds, right?
Yeah.
Actually, we have done certain studies in Tartu University using various polysaccharide materials to see the gelling property, water holding property, and viscosity of this material. Basic studies to see how a scaffold material will be, how stable the scaffold material will be. So based on the results from fucoidan or funeral samples, when we are modifying the samples, it have like excellent scaffolding properties. But we are yet to do the in vitro analysis to see how they are reacting with the cells, because normally in in literatures it is used as a combination with other existing material like chitos and andol, and since they are natural material, it doesn't have any cytotoxicity properties, but like at the same time modified furan and carrageenan products have shown excellent cell proliferating properties. So I'm pretty sure that since they have cell proliferating property, low cytotoxicity, and with excellent hydrogel forming property, this could be probably the future of tissue engineering scaffolds.
So another thing I would like to know is that you mentioned that they can be used in multiple cancer therapies. So are there any clinical trials ongoing with algae and cancer therapies? Are there any trials going on actively at the moment?
No, I'm not very sure about clinical trials, but there are like in vitro and in vivo analysis going on with algal products, which have shown like excellent anti-cancer, or basically they are good antioxidants. So this antioxidant property will somehow enhance the immunomodulating property. So this will be helping the normal cells to fight against them. But at the same time, the targeted cytotoxicity property towards cancer cell also observed in case of in vitro analysis in our studies. So when we tested against skin cells and colorectal cancer cells, they have shown like excellent cytotoxicity studies specifically towards cancer cell, but they were like kind of very stable towards normal cells. So I could say that
Okay.
yet the clinical studies will be completely different. They could be completely, but
Okay.
I don't know any of those. Probably they could be, but I don't know those.
Okay, thank you very much. Okay, one last question. You talked about the gels from algae. Do you have any experience of isolating these hydrogels from the like algae or?
Yep, we have isolated. I have isolated funoran from one strain, multiple strains of red algae during my first postdoctoral position in Tallinn University. So we used. What exactly is your question? Like,
I asked just if you have any experience with isolating gels.
I have used extraction process, and later we have done like some hydrogel preparation. Basically, they are similar to agar or carrageenan. So it has like this viscous property, and it could be if we are introducing any types of like what to say, you can actually modify the gelling property if you are introducing different elements to it. So basically by like changing the ionic properties of the metals or ionic properties of the materials in it, you can change the stability of the gel as well. So basically, initially, funoran will be like without any treatment, the funoran will be normally just viscous in nature. But like after treatment and after extraction process and after multiple treatments, this could make like a really hard gel, solid gel.
Okay. Thank you very much.
Oh yes, Alreza, please go ahead.
Thank you, Geetha, and thank you for the presentation and also the help that you are giving to us. Probably, I think we have already somehow had material from your algae, and we have somehow tried to get the extracellular vesicles, and it works.
Yeah.
It is, it is definitely we see, and also we see functionality. We see that they have different effects, and looking at your presentation, majority or nearly everyone is using algae itself. We are using the water that is left from algae culture. Then we are actually using the waste product of the algae culture there. And when I look at your presentation, looking at the big companies that are having that big fields with all the algae there, and so I'm just saying, God, how much extracellular vesicles are probably produced and are left in there. And we are coming to the point that okay, they have interesting potential applications or functions. Now, from one side, we are trying to understand, okay, how does it work and why extracellular vesicles are doing this? It is trying to understand the mode of actions and how that is taking place. It is a pure, potentially at the moment, scientific adventure. But at the same time, I was thinking, if we want to either bring it to market or even if we want to support our research, it would be good to work with the industry. And there are two industries that I can somehow think about. One is those that are producing algae in high quantities. Maybe they would be interested to somehow that if we tell them that, hello, we can use your wastewater and bring in another product for you, maybe they would be interested. We are doing this also with the plant producers. And the other thing is just, yeah, even that, or also trying to look at the downstream producers that, for example, use algae for, can use their products, the extracellular vesicles from algae in the beauty products, or they can use it in the, as antimicrobial products, or we see that effects is there. What do you think? How should we do? What should we approach? You are speaking about all of this and being in the algae world and so. What do you think for both of these? How can we do? Because the proof of principles, X months ago that we saw each other and we started this, that was at first we were thinking, would it at all work? Would that extracellular have any function? Now we see yes, they are functional, they do all of that. Now, how can we, in your opinion, we can bring it to the fruition of a potential work that can go further?
Thank you, Professor, for the question. I'm not an expert in like a commercial or like a large scale cultivation system, but still from my experience, what I understand is like for open pond or large open facilities, the contamination risk from different things will be very high. So if you're focusing on EVs specifically from my algae, probably you would be getting much more than EVs other than algae as well, like if you are focusing on open ponds or open systems. But in closed systems, there is a possibility to get EVs, as you mentioned. But at the same time, most of the algal systems, water will be recirculated. So basically, after the use, they will be disinfecting, cleaning the water, and then the water will be reintroduced to the systems again. So in that sense, collecting EVs should have like a separate system where it could be incorporated with the closed setup of the large algal setup. So you can collect the EVs during the harvesting process, and then later that water will be disinfected and used in the system. So it may need some specific technical support in the existing algal, if it's a closed loop system, in the existing facility. And forgot your second question.
No. Yeah, I was thinking about the companies there. Then, in your suggestion, those that are using closed systems would be the best ones to somehow approach them and somehow tell them that, okay, we have this. And yeah, since you are in the algae world, you are probably worked or know of these different companies that they use closed system, probably in Europe and this and that. What do you think? Do you think that they would be open to collaboration, cooperation? Or do you have any views on that? Or
I have undergone a training in Portugal with the A4F, one of the main company there in Lisbon. They have this open pond system and closed pond systems as well, closed loop systems as well. So also in Portugal, we have one of our collaborator from Azure. They also have like a company associated with university. They are mostly focusing on hematococcal strain. Probably they will be very interested in large scale EVs if you are looking forward to that actually. I could share the details probably in our next meeting. So
yeah,
that could be. But we have checked the hematococcal strain for the EV production. So that's another thing. But this A4F, Algae for Future, that's the company name, I think. They were collaborating with different academic institutes as well to customize algal products in a way like how they need it. So probably later in large scale facility, you can probably do that.
Okay. Maybe we need to have another meeting with you and some of us that we somehow sit down and see how we can get the...
I'm really sorry. I'm on maternity leave and kind of packed with multiple things. That's why I couldn't join the meeting, the last meeting.
Thank you very much.
So is there any more questions? Okay, so then let me go with my questions. I have quite a few technical related questions. Sorry for that. So the first one is like, when you grow the algae in your photobioreactor, do you add any antibiotics or any copper sulfate or any metal or minerals to improve their growth?
We grow them in specific media corresponding to each algae. So we do not add any antibiotics or antimicrobial agents. All the systems are sterilized, autoclaved, and closed. So previously we had like initially when we started the microagriculture, we had some contamination from the cyanobacteria, blue-green algae. We found like filamentous algae in all our cultures. Then we had to like sterilize the entire lab and start from the fresh culture again. So as far as we are like very careful and sterilize all the facilities and use the closed system and use filters for everything, for air, for any nutrient introduced after the closed setup, and do everything under the biosafety cabinet. So in that case, we can definitely control or regulate the microbial dimensions.
Okay, so in
do regular microscopic analysis to ensure that there is no other things other than microalgae.
So meaning that the conditioned media that you use, you know the exact constituents in that media. Okay, and then there's no copper sulfate or any such thing that...
There are copper contents, but each media has different components, so it will vary.
Okay. And the other thing is like, how did you check the cytotoxicity in fish? Like, is it the first one is like, how did you incorporate fish feed with the microalgae? Is it slow releasing, or is it just the powdered chlorella is converted into fish feed, or is it fish feed incorporated with chlorella?
Exactly. This part of the work will be done in Iceland by our collaborator, Metis. But the process is like we use different compositions of microalgae, combine it with the already existing feed composition. Basically, we are going to try like 10% and 5% of chlorella at the moment. We prepared the feed already, and the fish trials are ongoing. So it will be tested on salmon and trout fish models, and the toxicity evaluation will be based on the ex vivo analysis of the fish models.
Right. Okay, yeah, that's all for me. Is there any more questions to Geetha from the floor? Okay, if there's no more questions, I would like to say thank you very much. And we're so sorry that we have to disturb you during your maternity leave, but thank you very much for your participation and in sharing your knowledge with us. And we would like to see you in our future collaborations. And thank you very much for all the participants here today and joining with us and everything. Yeah, thank you very much.
Thank you for the platform. Thank you very much. Thank you, Dr.
Yeah.
Algae.
Bye.